synthesis is the process of linking amino acids using peptide bonds. This makes the polypeptide chain. In protein synthesis, there are two steps: transcription and translation. Transcription is deriving DNA into mRNA sequences, and then translation is using mRNA into proteins. It's making proteins.
At the start of initiation, transcription factors attach to the promoter region of the template strand. This creates the transcription initiation complex. Then, RNA polymerase II attaches to and binds to the template strand at the promoter region. It then begins to unwind DNA. The polymerase then begins RNA synthesis at the start point on the template strand. During elongation, the RNA polymerase II continues to move along the DNA strand while unwinding it. It reads sense DNA and matches it with correct mRNA nucleotides, adding to the mRNA strand.
In the 5 to 3 direction.
The termination of transcription goes to the termination sequence, then continues 10 to 30 bases this direction. When it reaches, it jumps off,
thus ending the pre-mRNA sequence.
RNA processing must occur before the pre-mRNA leaves the nucleus. And the modified guanine cap is added to the 5' end, while the poly-A tail is added to the 3' end.
Lysosomes contain snRNA. These snRNAs attach to the ends of introns. Some pull at the end of the intron so that these would get edited out. Therefore, the exons would then bind together so that pre-RNA is then edited into the real mRNA that it's supposed to be.
The mRNA coding dictionary is used to determine which. So translation is the actual synthesis of a polypeptide. It occurs under the direction of mRNA and takes place on the ribosomes.
Ribosomes facilitate the specific coupling of the tRNA to the mRNA codon. The tRNA anticodon couples with the mRNA codon during protein synthesis.
Ribosomes consist of two subunits, a large and a small, which is made by RNA polymerase in various proteins.
So at the A site, the tRNA enters carrying an amino acid, and then at the P site, the tRNA contains a growing polypeptide chain. Once it enters the E site, it then discharges to go off to find another amino acid.